So with that, I'm going to move on to our next presentation and introduce Julie Goddard. She is a professor in the Department of Food Science, and her research program focuses on understanding the interface between food contact materials, food, and microorganisms, and how to modify food contact materials to improve the quality, safety, and sustainability of our food supply. Julie, take it away.
Thanks very much, Martin. And I'm really excited to give a little introduction on my research program and some of the ways where I see we can use material science as ways to, as an intervention method towards reducing food waste and loss. As Martin mentioned, my group's overall research mission is in innovation in biomaterials and biointerfaces in food and agriculture. And the motivation for this work, food waste wise and beyond that, comes from just realizing how often our food comes in contact with materials on its journey from farm to fork, and how these materials, designed well, can have a big impact in improving food quality, reducing food spoilage, and if designed poorly, could have, unfortunately, the opposite impact. There's examples on the safety side as well, but there's an interesting example on food spoilage. There's an interesting case study, is that of soft rot bacteria, where contamination actually occurs on farm, but the spoilage indicators don't occur until downstream in the process. So that speaks directly to Martin's thoughts on the need for a systems level approaches, where you have a contaminating event happening here, but after the use of a lot of other resources to get a product to market and ends up not being saleable, there's a whole systems issue involved. There can also be cross-contamination occurring in many locations in the processing environment, from transporting to filling. Fouling of heat exchangers can reduce product quality. And we're becoming increasingly aware as well about the potential for cross-contamination in non-food contact regions, places like drains and ditches, where there's research showing that the same strain of persistent microorganisms have been traced in the same facility during decades-long studies. That can lead to spoilage and waste. And again, that spoilage and waste may not occur until additional resources beyond the product itself have been spent. So in my research group, we look at applications-driven research to improve food safety and reduce food waste, and identify new opportunities for byproduct valorization. And each of these research thrusts has application in material science solutions to reducing food waste. So in active food packaging, we're looking at using the packaging material to do more than just contain and protect the product, but possibly prevent light-induced degradation or just regular degradation processes that happen over a product's shelf life that make it go bad and no longer be saleable or good quality for consumption. In the biocatalytic materials area, I'm interested in using enzymes to take what might be considered a waste stream and upcycle or valorize it into value-added ingredients. And then finally, the area I'll talk mostly about today, which is in the area of antimicrobial and non-fouling coatings. And today, this is going to be the primary focus, and we're looking at these antimicrobial and non-fouling coatings as a strategy to reduce food waste by reducing the microbial spoilage organisms that might adhere to surfaces that become then points for cross-contamination onto foods and beverages. As you scour the literature for novel technologies, you can find some pretty cool stuff, right? There's fascinating examples that can be inspired by nature. Shown here are some really cool topographical approaches where we trap air at a hierarchical length scale to create a low surface energy and super hydrophobicity. And so there's an example of like the lotus leaf mimetic compounds and other kind of hierarchical nanostructures that can occur. We can also be inspired by shark skin, which is another biomimetic material opportunity where we can reduce frictional drag in swimsuits or ship hulls, preventing barnacle buildup on ship hulls and reducing fingerprints on our stainless steel appliances. And like I mentioned, the famous, probably one of the most famous biomimetic topographical approaches is in trying to recreate the lotus leaf. And there's some really cool stuff going on in the literature. But we do need to consider resources and scale. So this is a picture of the Cornell dairy plant, complete with the decorative cow tiles. And we have a range of topographies and base materials to consider: stainless steel, concrete, carbon, gasket material, et cetera. And important to remember, very low margins to play with, right? And this presents a unique challenge. And I think Caitlin mentioned this as well, like this whole moonshot idea, right? We need to innovate as researchers and scientists. I think of high pressure processing, my smartwatch. And I think if these ideas were stifled out when they were initially presented with some idea of this is not practical, it'll be too expensive, nobody's going to pay for it, we'd miss out on a lot of really great transformational technologies. But we can't forget that we are in a low margin field, right? We have to consider existing infrastructure and equipment, economics, et cetera. So balancing pragmatism and innovation, keeping both of those in mind in technology development to reduce food waste, whether it's a material science approach or any other, is I think essential to their success. So in this line of my research, we try as much as we can to integrate innovation and pragmatism. So the goal, long range goal, kind of the pragmatic thing, is to design a polymeric coating that can resist the adhesion, persistence, and cross-contamination of spoilage microorganisms that could cause food waste and loss. The goal is to have this coating capable of being applied to a number of different materials that might be used in food handling and production. And again, like I mentioned, we're particularly interested in the application in areas that are not routinely in a cleaning and sanitization zone, places like drain ditches, HVAC units, et cetera. And the idea would be to use a process similar to current coating processes, not a need for, putting in all new drains, putting in all new floors and things like that. So one particular polymer coating chemistry that we've explored uses a halamine, which you may have heard of. These are amines, amides, and imide functional groups that, when exposed to sodium hypochlorite, standard household bleach, can become reversibly charged with chlorine and enter an antimicrobial state. So here's an example of a polymer coating chemistry. If you zoom way in to look at its chemical structure, you might see something like this. And what's key here are these particular functional groups. These functional groups are capable of being chlorinated using the concentration of chlorine that's commonly used in sanitization of food contact and non-contact materials, 200 part per million bleach, right? And then they can become antimicrobial and halamines, like what's highlighted here. We've shown in our research that as you increase the degree of the coating, so the amount or thickness of the coating, you can then manipulate the microbial inactivation kinetics. And so we can see here with increasing number of bilayers up to 5, we're able to show a log reduction value that achieves its theoretical maximum or theoretical total kill in under an hour. And of course, the beauty in principle of these halamine-based coatings is that while they can lose their antimicrobial form upon exposure to high organic loads or when they're used to inactivate a microorganism, they can be readily regenerated over and over again by subsequent exposure to bleach, which is a typical part of routine cleaning and sanitization. So it enables a sort of boost to existing cleaning and sanitization regimens. And while there's some really interesting conclusions that can be found and reported with these coatings, the reality is microbes are persistent, especially in the nutrient-rich environment of food and beverage processing. And this is why we start to see the prevalence and persistence of the same fingerprint of microorganisms over long times in the same facility. Even the most beautifully designed antimicrobial surface or the most clever non-fouling material will eventually condition with protein and carbohydrates and lipids, the kind of building blocks of all of our food and beverage, right? And can be rendered a beautiful place for microbial adhesion and biofilm formation. And that's the trick, right? Is that if we only focus on an antimicrobial material where we expect that there will never be fouling by food components and never be buildup of microorganisms, that's not particularly practical. So while this surface performed very well in achieving a 5-log reduction of key spoilage and pathogenic microorganisms with broad spectrum efficacy, there was a need to expand on our original goal. We still wanted to design a polymeric coating that could inactivate microorganisms that contact it, but we also wanted to introduce a functionality to try to inhibit the initial microbial adhesion and facilitate the removal of bacterial biofilms that do adhere, sort of a non-fouling or easy clean surface. So to do that, we changed one component of the chemistry, and that's shown in these highlighted things here, these styrene subunits, to help reduce the surface energy so that the coating would also be non-fouling. We also, in doing so, made it so that there remained some cationic antimicrobials. So this is a little bit closer in performance and functionality to a quaternary ammonium compound. So this way we've got a dual mode antimicrobial character, so that even if the chlorinated halamines lose their chlorination, there remains some potential antimicrobial character through these cationic compounds. So shown here are some numbers. The key thing to keep in mind here is in developing a non-fouling coating, there's some well-established kind of numbers for surface free energy that make it so something can be considered a non-fouling coating. And that's that number shown at the bottom. So we can see that both polypropylene, which is a plastic commonly used in like buckets and things like that, has a very low surface energy and can be considered non-fouling. And then once coated with this material, even though we are introducing a number of polar compounds, we retain that non-fouling threshold. So that's a good thing. So then we characterized again the antimicrobial material activity of these materials. And we see that as prepared, the coating enables a greater than 3 log reduction in microbial populations compared to controls. And in this case, we're looking at things like just bacterial suspension on its own as a control, uncoated polypropylene, and then polypropylene that's been washed with bleach. And that's an important control to make sure that it's not just residual chlorine on the surface that's causing some character. And it is actually because of these functional groups that present the antimicrobial character. And we can see that when these coatings are chlorinated, we can achieve about a 6 log reduction, and even before they're chlorinated. So this is now the quaternary ammonium compound mimetic groups that are imparting an antimicrobial character. We can achieve a greater than 3 log reduction. And this is a good example of another gap that you can find between looking in the peer-reviewed scientific literature or standard methods and true applications. So this is a legitimate study that we did that leads me to always say, under the conditions of this study as performed in my lab, this was the result achieved. And that's important to note because you'll often see, you know, with a new technology, the whiz bang, hey, this is great, it does this, it kills that. You see a lot of times 100% reduction, which of course mathematically is not possible, right? But you can see these amazing 99.9% reduction of X, Y, and Z. That's great. But under what conditions of use, right? When my group, and honestly, the bulk of many other research groups that look at this class of antimicrobial materials, when we do these antimicrobial assessments, we pretty much always use as a starting point, buffer or water to do the antimicrobial activity assays. And we did that here in these results and the other ones that I showed. And that was the right thing to do, to follow the standardized established method for comparison. Yet testing antimicrobial efficacy in an aqueous solution is practically irrelevant to most systems, especially if you're considering, say, a trench drain in a food production facility. So this is just kind of another one of the points I want to draw back to is in new technical interventions to reducing food waste, material science or otherwise, designing an effective applications test is not trivial and it's not always easy. And sadly, it's not always done in the published literature or in the marketing kind of literature that's showing kind of the efficacy of a new treatment. But it's important. It's absolutely essential because in reality, materials are less likely to be exposed to this and are more likely experiencing something like this, right? Waste streams or food products full of complex constituents like dirt, proteins, et cetera, across a wide range of pH values with the introduction of alkaline detergents and acid sanitizers. So while it's important to start your proof of principle studies, as we did, in using a standard commonly used method, it's also important to challenge the material in the presence of organic matter. So we explored the performance of our coating as a antimicrobial material against conditions of increasing organic load. We used increasing concentrations of horse serum just to kind of mimic a wide variety of proteins and things that might be present. And it's a fairly standard thing to represent organic load. And we can see that up to about 500 part per million of horse serum, we're able to retain 2 to 3 log reductions in antimicrobial activity by the coating alone, and greater than 4 log reductions in microbial populations after dipping in bleach. Not surprisingly, as our organic matter increases, the ability of the halamine, the chlorinated groups, to have any efficacy drops, as with any chlorine treatment. As you increase organic load, it becomes less effective. But it was good to see that even with the introduction of a higher organic load, we retained a 2 or 1% or 1 log reduction of microbial populations afforded by the coated polypropylene compared to the kind of full growth of the control that had no material present. And you can learn a lot as well looking at confocal laser scanning microscopy images. First, ignoring the color difference for a moment, 72-hour biofilms formed on uncoated materials are thick, dense, evidence of a 3D morphology typical of a well-established biofilm. In contrast, on the bottom, we can see the coating when it's applied to polypropylene. We have a much reduced cell density and appears as more individual cells rather than a thick kind of 3-dimensional biofilm mass. Taking into account the live dead staining, where red is dead or non-viable or membrane compromised and green are living cells, we also see that the bulk of the cells in the native polypropylene are viable, the uncoated material, membrane intact cells, whereas in the coated material, the bulk of the cells that do adhere are actually non-viable. So this is a big finding, right? Because it shows our hypothesis from earlier might hold some water, that even your best antimicrobial non-fouling material, given enough exposure to good, nutritious environment, will result in bacterial adhesion and potentially biofilm formation. So if those adherent cells could be largely non-viable, that's really good. We've also shown that in addition to reducing the amount of viable biofilm that forms, we've made it easier for those biofilms to be removed using a simple water rinse. And this goes a long way as well in reducing the transfer of persistent food spoilage bacteria from hard to clean growth niches like drainage ditches, while perhaps even reducing the amount of chemical cleaners and sanitizers that we're reliant on using to clean these regions. So as I begin the wrapping up here, I want to briefly go back to the idea of developing material science solutions to reducing food waste that bridge both innovation and pragmatism. Bring your attention back to the chemical structure of this coating that we were using in some of these experiments. These cationic groups enabled our materials to have dual antimicrobial activity, which was important, especially as we increased organic load. But it brings a good question of, wouldn't these cationic groups actually promote fouling on a surface, right? A lot of the biological and food world is negatively charged. And what you don't want to do is make a positively charged surface to which there will be adhesion of proteins and things that essentially make a beautiful place to form a biofilm. And this is the first of 3 seemingly boring, but tedious and time-consuming experiments that are very important in applications-driven research. So in this case, interfacial pKa, we are looking at whether or not there is a net charge. So if there were a net charge, if you come back to chemistry, intro chemistry, remember that there would be an inflection point, right? Whether you're analyzing using contact angle as we are here, or some titration or something else. If there is an inflection point, that marks the pH value at which there might be a change from protonated to unprotonated, charged to uncharged. So the fact that we're looking at flat lines, aside from the fact that this is, I don't even know, thousands of data points collected by a student, it basically tells us there is no net charge on the surface. That's a big deal in terms of showing that there's unlikely to be a conditioning layer that could then promote fouling. I've shown you some exciting micrographs, and they're fun to look at. This is a boring one, but it's actually really important because we want to see a uniform coating. And it highlights the importance of another application-driven science point, right? Statistically sound image analysis, making sure we're using samples that represent the population, appreciating and reporting what is seemingly boring data, but is absolutely essential to know if a particular intervention might be appropriate in a given application. And these application studies don't always feel glamorous. In this study, we exposed a coating to 168 cycles of alkali detergent exposure at the recommended, temperature and concentration and cycle time, as well as acid sanitizer. We've done mechanical abrasion studies, exposed things to the temperature extremes they might see. These are time-consuming studies. But again, what we're looking to see is with additional exposure to pretty rough environments, are we maintaining a surface chemistry that indicates that our coating is intact? And that's another thing that's pretty important to consider when we're looking at applications of material science solutions in reducing food waste. And as absolutely necessary as these kind of boring research projects are, not projects, but boring graphs are for the importance of having something that's industrial translatable, I don't want to leave you with a bar graph to remember me by. So here's a false colored SEM, fresh off the presses from my PhD student, Autumn Rundlong, where we're exploring a new type of antimicrobial non-fouling coating in which polyurethane chemistry is modified to present antimicrobial and non-fouling character. So a couple key takeaways as I wrap up here. We've shown that antimicrobial and non-fouling coatings can reduce microbial persistence on surfaces. Now, there's applications of this type of work in food safety as well, but we're increasingly knowing the importance of reducing microbial cross-contamination in terms of reducing microbial food spoilage. And not all antimicrobial agents perform well after being bound to a polymer structure. Sometimes they might change their performance depending on the pH value they're exposed to or exposure to other extreme environments. But I think for this type of work and a lot of other interventions, keeping an eye on both innovation so as not to lose out on a possible really cool shoot for the moon idea, but also considering end use application and pragmatism when possible, I think is critical for the success of new technologies. Ongoing, we're looking at scalability, looking at how can we make chemistries that are more scalable and something that could be much more adoptable by end users. And as always, we're interested in effective application studies as much as we are interested in cool new polymer chemistry techniques. With that, thank my group, thank FAR for supporting this work and some of the other funders, and I'm happy to open up for any questions.
Thanks, Julie. Any questions for Julie? Please type them either into the chat or you can raise your hand and we can unmute you to ask the questions. So here's one question. Can these coatings, either the ones you described or any others, be done in field with existing equipment, or does it need to be done at the time of manufacturing? So I guess the question is, can you retrofit existing surfaces with some of these technologies?
That's a great question, and one of the things that in recent years we've been really striving for. The goal would be absolutely it can be done on existing equipment, possibly even equipment that had previously had some other coating, right? That you could then during a shutdown apply this coating instead of a different coating and then have some additional benefit from it. And there's different approaches as well, right? So depending on the intervention or the location, say we wanted to produce a gasket material, right? Where the gasket presented some antimicrobial or non-fouling character. In this case, since gaskets are routinely replaced, it could make sense for that to be something that, it's the whole material that gets replaced and you work with a gasket manufacturer. But for the case of coatings for drainage ditches or floor coatings, you'd want that to be something that you could, purchase this material instead of kind of a material that had similar mechanical properties, but without the antimicrobial non-fouling character. a great question.
Okay, thank you. Any other questions or comments? One other question.
Regulatory approval, yeah. This is the question, and it's a great question, and it's particularly brings up the fact that it might be different in different countries, and it probably is, right? So there's regulatory approval for food contact materials. Food non-contact materials would be a slightly different process. And how complex and time-consuming is it, I think, depends on how new the structures are compared to others that are already approved in the United States. The Code of Federal Regulations has a listing of materials that are already approved and constituents of materials that are already approved for food contact use. Food non-contact use, of course, has its own level of approval, but there's a process that's pretty common, and any new material used, whether it's for food contact, food packaging, or non-contact, has to go through that process. So it's a fairly standardized process.
Okay, any other questions for Julie? Okay. Having none, we will take oh, there's another one.
Autumn, yes, great. So the student that provided that fabulous last SEM there reminds me that the EPA in the United States, the Environmental Protection Agency, also has to have approval of antimicrobial compounds, whether they're used in sanitizers or in polymers. So that's a great point, Autumn. Thanks for bringing that up.